A PhD's secret weapon, the 4 biomarkers every expert should, but doesn't analyze. These are the most useful tests and are the ones that no one ever orders. This video and podcast is available for free for 48 hours. After that, it is available only to MasterPass members. So check out the description for how to access it for all time. Everyone in the health space says that they want to get to the root cause. Then they order lab tests that barely scratch the surface. Everyone in the health space says they want to support mitochondrial function, but they ignore the main fuels of the mitochondria when running labs. Measuring the markers discussed in this video, podcast, and article, depending on which one you want to consume this content as, can help uncover whether you are taking too many supplements, overdosing on the wrong one, or the wrong ones, eating the wrong diet, or whether you're succeeding or failing at addressing your own idiosyncratic bottlenecks in energy metabolism that limit your wellness, your performance, and your longevity. Everyone in the health space wants to optimize blood glucose, but no one is putting glucose into context. No one is looking at what the glucose is turned into, and no one is looking at what it is competing with as a fuel. And when people do look at the other fuels, they do so with false assumptions. For example, lactate is for exercise. Ketones are for keto diets. Wrong and wrong. And the poor victim that always gets ignored is the redox status of these molecules, which may sound like a super nerdy concept, but it's actually the single most important thing you can understand if you want to get to the heart of why your mitochondria are not performing at their best. I have a client whose practitioner put her on keto and high-dose thiamine to solve dysautonomia, which probably isn't even dysautonomia to be honest, which made her 5 times worse and bedridden. Had anyone ever tested whether thiamine raised or lowered her lactate, this never would have happened. And yet, if anyone looked at the ratio of lactate to pyruvate and the ratio of the 2 major ketone bodies, beta-hydroxybutyrate and acetoacetate, one could quickly determine whether the most important mitochondrial fix for any given person would be thiamine, B6, or biotin on the one hand, or riboflavin, CoQ10, or copper on the other. And the craziest thing is that sometimes these nutrients are needed in isolated megadoses. So not CoQ10, copper, and riboflavin, but CoQ10 or copper or riboflavin to address an otherwise extremely difficult to find genetic idiosyncrasy in a manner where traditional measures of nutrient status, even the panels that I prefer as the most well-validated markers of nutritional status, would never help you because everything's normal. All right, what follows is only available for free for the first 48 hours. After that, it is only available to MasterPass members. So if you want to consume this content in an enduring manner, if you want to take notes, if you want to come back to it, or if you missed the 48-hour mark, check out the link in the description for how you can access the video, the podcast, as well as the full written article that is fully referenced and links out to all the supporting information. What you're going to find in here is, what do these ratios tell you? What are the many causes of elevated lactate? What are the confounders that you need to look for, especially if you're measuring lactate at home and can't measure pyruvate? When to narrow in on nutrition-responsive idiosyncrasies of energy metabolism, the normal biochemistry and physiology of lactate, pyruvate, and ketones, the normal ranges in blood and urine, the stability of lactate, pyruvate, and ketones in blood and urine, very relevant to how to take care of your samples for a test and whether the tests are valid or not. And then the big one that you really want to understand, and in fact, if you just want the practical stuff, you can just skip to this part. It's how to interpret the results. Potential uses of thiamine, riboflavin, B6, biotin, copper, CoQ10, low-fat diets, ketogenic diets, near-infrared, fasting feeding resets, methylene blue, PQQ, acetyl-L-carnitine, creatine, high-dose vitamin C, and MK4, and other things. And then at the end, we're going to put it all together into a practical unit, an algorithm of sorts. Okay, so let's start. What do these ratios tell you? So the first 5 sections we're going to go over now explain how these ratios can be measured and used in broad strokes to conveniently collect the major themes in one place. I would very much recommend that you consume the full article before acting on any information in these sections. And also, always keep in mind that I am not a medical doctor. This is not medical advice. You are not my patient, and you should never implement anything until you A, understand it in full, and B, discuss it with your doctor. Okay, the biochemical principles at a glance, informational, educational only. The biochemical principles at a glance. These are the principles involved. So electrons taken from glycolysis, which is how glucose is broken down, or how carbohydrates are broken down, are transported from the cytosol to the mitochondrion using the malate aspartate shuttle. In the mitochondrion, they have their energy extracted by the respiratory chain to use for the synthesis of ATP. If the breakdown of food energy exceeds the activity of the respiratory chain, electrons build up. As a release valve, the electrons are loaded onto pyruvate to form lactate in the cytosol. The cytosol is the main compartment of the cell. If you think of the cell as a many-roomed house, the cytosol would be like the hallway, and are loaded onto acetoacetate to form beta-hydroxybutyrate in the mitochondrion. The mitochondrion is usually called the powerhouse of the cell. You could think of it, if you're thinking of a many-roomed house, it would be the it would be the part of the basement where the burners are and whatever, I guess. But you can think of a mitochondrion as an organelle. An organelle is like a room in a house. The cytosol is like the hallway that connects all the rooms. You could think of it like that. And of course, if you're thinking of functionally, what do they do? The mitochondrion is the powerhouse of the cell. Okay, so the release valve in the cytosol is the conversion of pyruvate to lactate, and the release valve for the extra electrons in the mitochondrion is the conversion of acetoacetate to beta-hydroxybutyrate. If the lactate to pyruvate and beta-hydroxybutyrate to acetoacetate ratios are high, that means that the rate of catabolic breakdown of food molecules is exceeding the rate of the respiratory chain. If the two ratios are low, the rate of the respiratory chain is exceeding the rates of the catabolic breakdown of food molecules. So you can think of these as two steps in an assembly line, where the first step is the catabolic breakdown of food molecules, and the second step is the utilization of the energy of the electrons taken in that process in the respiratory chain. If the two ratios are saying the same thing, the malate aspartate shuttle is working. If the two ratios are saying the opposite thing, the malate aspartate shuttle is not working. Now, from these, we can harvest all kinds of nutritionally actionable pieces of information, and we will get to those soon. The normal ranges. These are my best estimates from the literature with varying levels of confidence, justified in detail later. All apply to an overnight fast, and you should always abstain from alcohol for 48 hours before testing. So a blood lactate to pyruvate ratio, the normal range is 4 to 16. A urine lactate to pyruvate ratio, normal range is 5 to 10. Blood beta-hydroxybutyrate to acetoacetate is 0.5 to 3, with less than 0.3 being strongly out of range, greater than 8 being strongly out of range on the high end. And if you're on a keto diet, these are altered so that the normal range extends upwards to 8. So in other words, if you're not on a keto diet, you could say the ratio could be 0.85 to 3, but if you're on a keto diet, it would be 0.85 up to 8. And if you're above 8 or you're below 0.3, you are strongly out of range. The middle of that range can be ambiguous because there can be, it's not as if you are binarily on keto or kicked out of keto, but keto is a spectrum. So if you're in that range, so basically less than 0.3 or greater than 8 are ratios that are just reliably out of the range. Whereas in the middle, whether it's too high or too low, you have to take into context the carbohydrate and protein content of your diet to really say, like, how keto should I be in this context? Because the more keto you are, the more reasonable it is for your ratio to go from 3 towards 8. Now, the urine beta-hydroxybutyrate to acetoacetate ratio, if you're not on a ketogenic diet, should be between 0.65 and 2.0. If you're in light to moderate ketosis, it could be 0.2 to 6. And if you're in very deep ketosis, the normal range could go up to 12. And so again, less than 0.65 or greater than 12 are pretty obviously out of the range. That middle area is a gray zone depending on how keto are you. Now, only relevant to what we will be calling pattern 3 below we haven't gotten there yet. A pyruvate over 0.17 millimoles per liter is high. Those are the normal ranges. Now, these are the patterns to look for. Pattern 1. If the lactate to pyruvate ratio is high, but the beta hydroxybutyrate to acetoacetate ratio is low, this suggests a deficiency in one or more components of the malate aspartate shuttle. This includes genetic disorders that are treatable with ketogenic diets, vitamin B6 and serine, treatable with those three things, or biotin. It may also include deficiencies of biotin or B6, meaning nutritional deficiencies, not necessarily the lack of a nutritional fix for a genetic issue. Pattern 2, if both ratios are high, this suggests a respiratory chain impairment. This includes genetic impairments that are treatable with high-dose riboflavin or coenzyme Q10, acquired disorders caused by drugs and herbs such as metformin, berberine, and ciprofloxacin, or Cipro, the drug that causes people to get floxed. It may also include copper deficiency. This pattern may also indicate potential roles for ketogenic diets, low-fat diets, high-dose vitamin C and K, methylene blue near infrared, or stimulation of mitochondrial turnover using various drugs or using my fasting feeding reset, all depending on the specific subtype of the respiratory chain impairment. In other words, this isn't a bucket where the bucket of those things helps. It's a bucket of different types of impairments where different things in that bucket may help, if that makes sense. Okay, pattern 3 is high pyruvate, normal or low lactate to pyruvate ratio. Lactate is usually elevated but doesn't need to be for pattern 3, and this would suggest a genetic impairment that could be treated with a ketogenic diet or thiamine in high doses. However, simple dietary deficiency of thiamine is not likely to cause this pattern. If there's more than one underlying problem, these 3 patterns can combine into 4 additional patterns in the way that the 3 primary colors combine to form 3 secondary colors, or can all be mixed together to make the color brown. This diagram that's being shown in the video and is shown in the article, so if you're listening to this on a podcast, you might want to check out the video or the article to see the diagram, is going to be discussed and justified later, but is placed here for general convenience. And so what this is saying is you can, if you make malate aspartate shuttle the red zone, a respiratory chain impairment the blue zone, and pyruvate dehydrogenase the yellow zone, those are the 3 primary colors, and you can have the red and yellow zones mixed to make orange. You can have the blue and red zones mixed to make purple, and you can have the blue and yellow zones mixed to make green, and then you can have all of the colors mixed together to make brown, and that leaves us with 7 different patterns that we can take they include the original primary colors, the secondary colors, and the brown. And so this complicates matters because if there's 3 primary patterns, you need to be more discerning to understand the more complicated patterns that emerge from the combinations of those patterns. All right, and that's what we're going to try to get down into detail here. So the way that I view these, I'm not a medical doctor. I don't diagnose diseases. I don't treat diseases. I'm not looking for disorders. I'm just trying to understand what we know about biology, and I include the study of disorders in order to understand the normal function of nutrients and where things can go wrong in a way that we can optimize in the absence of a diagnosable, treatable disorder that a doctor should work with. And so the way that I look at this is that we can use these markers, especially the ratios of the 4 markers, so the lactate to pyruvate ratio and the beta hydroxybutyrate to acetoacetate ratio, which we could also call the ketone ratio, to look at the use of thiamine, B6, or biotin, and how these could be imbalanced with riboflavin, copper, or Q10. This could be manifested as, just as a general example, by a failure to fix a high lactate to pyruvate ratio when the ketone ratio flips from low to high. And if you look at the diagram, this would be signified by seeing someone move from purple to blue. And so we can expand on any of these mixed conditions and we'll go in detail through each one, but this is just an example. So at the beginning, I had said that a client got worse with high-dose thiamine and the ketogenic diet. So this, clearly, in this case, the practitioner thought they had the same problem and had recovered with high-dose thiamine and a ketogenic diet, and made the grossly ridiculous assumption that because they got better from one problem with one solution that their patient would, which if you are a normie sounds totally reasonable, but if you understand idiosyncratic bottlenecks in energy metabolism, the fact that everyone has them, then it's insane. And I mean, it plays out as insane because it actually made the person 5 times worse. So in that case, especially with the work that we've done together, it's now become exceedingly clear that what happened was this, well, what happened, what is very clear is this person has a combination of, so this isn't exactly the issue that's shown in the diagram. This person, because the assumptions of the practitioner were thrown on top of it, right? So had I been looking at this person, what would have happened would have been that the lab work would have first revealed that the primary problems were a combination of a fatty acid oxidation disorder and a respiratory chain impairment. It's very possible that had I done the lab work in sequence in the way that I actually did, in which I no longer do in the BioOpt Health program, now everything is done at once, it's quite possible that I could have made this person worse because the riboflavin supplementation would have helped fix the fatty acid oxidation problem, put stress on the respiratory chain, and then that would have made the lactate to pyruvate pop up and the beta hydroxybutyrate to acetoacetate ratio pop up, manifesting that issue. And then I could have said, aha, this is why the riboflavin is making some things better and some things worse. And then we would have addressed the respiratory chain issue. What actually happened was the practitioner was taking assumptions from their experience, putting the person on a ketogenic diet and high-dose thiamine. The ketogenic diet worsened the fatty acid oxidation disorder, and the thiamine worsened the respiratory chain disorder, because both of these things are delivering now more electrons to a burdened respiratory chain. And at this point, we've done the full level of lab work, so we've actually done a functional test of the respiratory chain. We have a very good handle on this, but all of this could have been evaluated from the very beginning by seeing the lactate and the fact that certainly the thiamine at that point would have increased the lactate, and that's how you would have discovered it without having done most of those labs. Okay, so the logistics involved here. The Great Plains Organic Acid Test is the best way to get both of the ratios in the urine. Quest has both beta hydroxybutyrate and acetoacetate in the blood. Quest has the lactate to pyruvate filtrate test. And it's important to abstain from all alcohol for 48 hours prior to testing. I don't think this is necessary, but if you're worried whether the lactate test would capture intestinal bacterial lactate production, Quest does have a D-lactate, which is actually very expensive if paying out of pocket, and you could use that to confirm or refute it. I don't think it's necessary. Home tests that could be of value would be Anova Biomedical Lactate Plus for finger prick lactate and Keto Mojo for finger prick glucose and beta hydroxybutyrate. And it's important to realize that you cannot capture the pyruvate or the acetoacetate at home in the blood, and you can't compare blood and urine. So you can't measure beta hydroxybutyrate in your blood and urine acetoacetate and take that ratio. So the best thing to do is to take them serially, inclusive of the day that you also go get the Quest tests and that you get the Great Plains test. So therefore you can corroborate your home, you can calibrate your home measurement to the one that you cannot do every day, or the two that you can't do every day, which is the Quest measurements and the Great Plains measurements. Great Plains actually is now called Mosaic. And so what this will allow you to do is to say, well, my lactate is generally increased by X, or my lactate is always high in the morning. Does this correlate with a high lactate to pyruvate ratio or not? And that's valuable to know for reasons that we'll go into detail in a bit. You want to do these measurements in the resting state, so you should be relaxed at rest and without exposure to extreme temperatures or any other stressful stimuli. Similarly, your hand and forearm that the blood is drawn from should be similarly relaxed and at rest as you are yourself. The urine samples should be refrigerated immediately after collection and fully frozen before shipping, and shipped with a fully frozen ice block in an insulated package to prevent thawing in transit. These things are generally covered in the kit instructions, but still be very mindful of them. The blood processing, so the blood samples for the ketone ratio do not require special processing. However, when the blood is drawn for the lactate to pyruvate ratio, the phlebotomist has to have ice cold perchloric acid in an open tube ready to be mixed with the blood immediately after drawing. It must then stand for 10 minutes before centrifuging. The person getting the test done must be informed of this, print out the instructions of the lab, show them to the phlebotomist, and verbally confirm that they have the perchloric acid, must know it has to be mixed immediately before centrifuging. Otherwise, there's a high likelihood of lack of familiarity on the part of the phlebotomist that will ruin the results. In my experience, you probably don't want to call them and ask because they probably don't even know, and you want to go there and insist that your doctor told them that they go look for the perchloric acid and it's probably in the freezer. I'm serious. So urine versus blood, I have higher, I'll say one more thing. The Quest instructions say that you can't wrap that you can't wrap up the arm for the blood draw. That actually doesn't matter. It only increases lactate if it's like 20 minutes that it's wrapped up for. But so it's exceedingly important to mix it with the perchloric acid in the right volumes and let it stand for 10 minutes before centrifuging. It doesn't matter what they say about wrapping the arm. Urine versus blood. I have higher confidence for the blood ranges than the urine ranges, and the normal kidney handling of lactate suggests that blood will more faithfully communicate metabolic changes than urine, specifically for the lactate to pyruvate ratio. So the urine ratio should be used as one piece of information that may prompt blood measurements, not as a confirmation of a metabolic abnormality. Even if the abnormality is not found in urine, strong symptomatic suspicion justifies a blood measurement. Remember, we're not talking about diagnosing disorders here. We're just looking for gaps in optimal biochemistry. Let's take me as a case study. So earlier this year, I worked my way up from 600 micrograms per day of biotin. This is at the beginning of the year. 600 micrograms per day of biotin to 10 milligrams per day. And I supplemented 10 milligrams per day in 2 divided doses for approximately 5 weeks. The biotin was taken on an empty stomach as far away from my most nutritious meals and my other supplements as possible, with the expectation that it could therefore not impact my absorption of pantothenic acid or lipoic acid, because these 3 nutrients compete for each other's absorption. My urinary pantothenic acid on the Great Plains test was unchanged, and the Vibrant America concentrations also suggested that this had no effect on my lipoic acid or pantothenic acid status. So I went off biotin for 9 days to test my basal serum biotin, and then entered a biotin maintenance period of 600 micrograms per day. The biotin loading raised my serum biotin level from 2.5% into the normal range to 35% into the normal range. And prior to my experiment with biotin loading, my Great Plains organic acid test showed that my lactate to pyruvate ratio was 13.3, while my beta hydroxybutyrate to acetoacetate ratio was 0.16. The lactate to pyruvate ratio was high, but the ketone ratio was low. This is consistent with pattern 1, an impairment in the malate aspartate shuttle, and is extremely consistent, or it seems so, with my biotin-centric hypothesis that the main abnormalities in my metabolism are from pyruvate carboxylase deficiency, secondary to a deficiency in recycling utilizing biotin. And I described these abnormalities in self-experiments in the biochemically unoptimized state last year. I have now revised this substantially to completely ditch the biotin-centric hypothesis. But let's keep this in the context of what it immediately appeared to do and what immediately appeared to make sense after this experience. So before I started biotin loading, I had identified a heterozygous mutation in ACAD9 with a frequency of 0.8% and conflicting interpretations of pathogenicity. I've since identified actually that there's 2 of them. The whole genome sequencing doesn't allow me to know whether they're in the same side, like my mother versus my father. But it's possible that I'm compound heterozygous or that I just have the 2 impacting one of my ACAD9 genes. I can't tell, but I do have 2 rare, possibly pathogenic mutations. So these lead to a combined fatty acid oxidation disorder and a respiratory chain disorder that's treatable with high-dose riboflavin. I dismissed the relevance of this mutation partly on the basis that a respiratory chain disorder or impairment should lead to an elevated ketone ratio, whereas mine was low, and partly based on seeming incompatibility with my acylcarnitine profile. However, it did occur to me that the deficiency in the malate aspartate shuttle could mask a deficiency in the respiratory chain, and that the evidence for this would be a failure to lower the lactate to pyruvate ratio and to cause a flip in the ketone ratio from low to high. So the first health effect of biotin supplementation was a nearly immediate and completely unmistakable increase in nocturnal penile tumescence, which normal people call morning wood. And the biotin also improved my subjective sense of how easy it was to fall asleep at night. However, biotin also increased my fasting lactate and my blood glucose. And by the end of the biotin loading period, I had developed minor short-term memory loss and clumsiness. Once or twice a day each, I would forget why I'd walked into a room, and separately I'd drop something for no reason, and I had become easily angered in general. Biotin also worsened my alcohol tolerance, where less than 1 glass of wine caused my hands to start tingling, and also drove a much higher than expected rise in my finger prick lactate. While trying to maintain a lower dose of 600 micrograms per day after this, I felt tired. On the morning of my last day of high-dose biotin, my ketone ratio on the Great Plains test had flipped to 1.56. This represents a 10-fold increase from low to the higher end of the normal range. The same morning, my blood lactate to pyruvate ratio as measured by Quest was 23, which was flagged as high by Quest, which uses a cutoff of 20, and it's even higher by my standard of 16. Quest detected 0 acetoacetate in my blood. If I impute half the limit of detection for the acetoacetate, my ketone ratio is 5.9, which is nearly double the top of the normal range I personally derived from the literature. So the biotin flipped me from pattern 1 to pattern 2, which is consistent with a malate aspartate shuttle defect masking the impact of the riboflavin responsive respiratory chain disorder. Biotin resolved the malate aspartate shuttle defect, allowing trapped electrons to move from the cytosol to the mitochondrion, but these overloaded the respiratory chain, causing a rise in both ratios and a transition to pattern 2. Using the color diagram, again, if you're listening to the podcast, please go to the video or the article. But using the diagram, I initially thought I was in the red zone because that's what it looks like when you're in the purple zone. It looks like the red zone and it fools you. But I was actually in the purple zone, and the only way I could learn this was by pushing myself into the blue zone by fixing the problem in the red zone. Now, I want to say that since I wrote all this up, I've now come to the point to believe that the ACAD9 is the overwhelmingly single most important thing, and that the biotin stuff really doesn't matter. That biotin and pyruvate carboxylase versus riboflavin, which is allied with transamination of glutamate to alpha-ketoglutarate and alpha-ketoglutarate dehydrogenase feeding glutamate into complex 2, that these 2 things are in anaplerotic competition, and that because of the ACAD9 mutation, I actually do not want pyruvate carboxylase activated, and I want things to flow primarily through complex 2, and I want to fill up my citric acid cycle primarily through glutamate and to some degree through succinate, and that it's the filling up of the citric acid cycle that drives all of the benefits that I got from biotin, such as falling asleep earlier and massive increase in morning wood. This is all driven by anaplerosis, or the filling up of the citric acid cycle. And biotin was only one path toward that end. And because of the ACAD9, biotin and pyruvate carboxylase are not the path that optimizes me. So I can get those benefits simply by optimizing around the ACAD9 and optimizing around feeding things through complex 2. And that can then also get me massive benefits. All of these will be covered in the future in much more detail on my own case. Here, I'm simply using my case to demonstrate how with the color diagram, someone can be in the purple zone and it can look like they're in the red zone until you fix one of those problems and suddenly they look like they're in the blue zone. So in other words, having a respiratory chain impairment combined with some impairment in the catabolic breakdown of food molecules will mask the respiratory chain problem. And you basically have two options. The one that I prefer now is to run all the labs that are relevant first, so we don't have to figure things out sequentially, which is what I do in the BioOpt program. But my purpose here is that if you want to keep things simple, you can simply monitor lactate, glucose, and ketones at home while you serially test the two ratios in the lab on an as needed basis or serially before and after interventions. And then you can take a little bit more time to figure it all out, but you can figure it all out quite rapidly. And you can have real-time measurements in home where you try to fix something that looks like a thiamine problem, but your lactate goes through the roof, and now you have a heads up that you're either making something worse or you are fixing one problem and revealing another. And this can rapidly tune you in to make sure that you don't push things too far when you are pushing yourself in a very accidentally pushing yourself in a very negative biochemical trajectory. So interestingly, a caveat to my own measurements were that my urinary lactate and lactate to pyruvate ratio on the Great Plains test, or now Mosaic test, actually went down. And I don't have any preloading blood tests from Quest. However, my waking finger prick lactate that day was 2 millimoles per liter, which was up to that point my highest morning lactate ever. The only thing that's ever pushed my morning lactate higher than that has been eating maple syrup due to a fructose issue, but that's separate. So my finger prick lactate had been increasing the entire time. And had I paid more attention to it, and had I not been stuck in my biotin-centric hypothesis, I could have stopped that or eased back on it a lot earlier. But in any case, I think the blood results for the lactate to pyruvate ratio more faithfully reflect the physiological processes of interest. And I believe this is best explained by the kidney having a greater oxidative reserve than the liver in the fasting state, and having a stronger propensity to use even extremely high concentrations of lactate for energy or for gluconeogenesis. And I'm going to explain these principles further in the sections below. So withdrawing the biotin made the short-term memory loss, clumsiness, and quick temper disappear. It also caused my blood glucose and lactate to go back to normal. However, going more than 2 days without 600 micrograms of supplemental biotin caused the morning wood to go away. But remember, I have now decided that there are two paths to the morning wood. One is pyruvate carboxylase, and one is feeding glutamate through glutamate transamination to alpha-ketoglutarate and alpha-ketoglutarate dehydrogenase. And these two things are both anaplerotic. All I need is citric acid cycle anaplerotic to get decreased sleep latency and increased morning wood that I got from biotin. I don't need biotin for it. And so at the time, I had concluded that originally I needed a maintenance dose of 150 to 300 micrograms of biotin per day. But I'll tell you now that since I've gotten rid of the biotin and have focused on the riboflavin feeding complex 2, I've increased my calorie intakes from 2,400 calories a day to 3,500 calories a day to prevent myself from leaning out too much. So take that for what you will, but I'll be covering that later. Not in this video podcast, but in a future article. All right, enough about me. Let's get into the science. So first of all, elevated lactate has many causes. Anything that causes lactate production to exceed lactate clearance will raise the concentration of lactate in the blood, and usually the lactate to pyruvate ratio along with it. Typically, this is a result of decreased delivery of oxygen to one or more tissues, decreased utilization of that oxygen in mitochondrial metabolism, or a shift towards glycolysis. However, a shift towards glycolysis will, if it's not accompanied by a deficit in the supply or utilization of oxygen, will generally elevate lactate along with pyruvate and therefore not change the ratio. So for example, adrenaline and cortisol should increase glucose, increase pyruvate, increase lactate together because glucose is being mobilized to be burned for energy and all aspects of that process are going up. By contrast, anemia should cause tissue hypoxia, which will increase the lactate to pyruvate ratio. Insulin should lower blood glucose, increase pyruvate and lactate together, but in the presence of healthy mitochondrial function, it should not raise the lactate to pyruvate ratio. Insulin also suppresses ketogenesis, but should not do anything in particular to the ketone ratio. In diabetes, the lack of insulin signaling causes glucose and ketones to rise, but does nothing in particular to the ratio. Thus, during diabetic ketoacidosis, lactate is moderately elevated at 1.6 millimoles per liter, but the lactate to pyruvate ratio is normal at 11.1. The ketones are sky high with a total over 10 millimoles per liter, but their ratio is normal at 3. Alcohol raises lactate, and especially when used without carbohydrate and protein, is ketogenic. Each molecule of alcohol generates one NADH in the cytosol and one in the mitochondrion. I described the biochemistry behind this in more detail in Why Alcohol Raises Lactate, a section of my self-experiments in the biochemically unoptimized state. For healthy people drinking several drinks in one night, a rise in blood lactate to between 1.5 and 2 millimoles per liter is normal. The peak lactate is 16 hours after alcohol consumption. It can be as high as 3 millimoles per liter, and lactate can stay elevated for at least 20 hours. Therefore, it is strongly recommended to abstain from all alcohol for 48 hours prior to having blood drawn for lactate at a lab. In alcoholic ketoacidosis, the lactate to pyruvate ratio reaches an average 19, and the beta hydroxybutyrate ratio reaches an average of 7. This is consistent with pattern 2 and is a result of drinking huge amounts of alcohol while otherwise being malnourished, leading to the accumulation of an NADH load that cannot be cleared by the respiratory chain. The effect of cancer should be inconsistent. Cancerous tissue may shift toward glycolysis due to the Warburg effect, but they do so locally. How it impacts the lactate and pyruvate in the filtrate of whole blood would depend on how much of each substrate is escaping into the general circulation. Here is a list of causes of elevated lactate: high-intensity exercise, cardiogenic or hypovolemic shock, advanced heart failure or severe trauma, sepsis, hypoxemia, which generally needs to be severe with a partial pressure of oxygen under 30 milligrams per millimeters mercury to rise lactate to acidosis levels, carbon monoxide poisoning, seizures, shivering, diabetes, cancer, liver disease, pheochromocytoma, a usually non-cancerous catecholamine-secreting tumor of the adrenal gland, cocaine, propofol, which is an anesthetic, reverse transcriptase inhibitors that are used to treat HIV, beta-2 agonists used to treat asthma and COPD, or chronic obstructive pulmonary disease, methanol, ethylene glycol, diethylene glycol, and propylene glycol. These generally require industrial exposure at high levels, but sources of small amounts of the glycols include antifreeze, paint, ink, plastic, lubricants, cosmetics, and other personal care products. Methanol is used as a solvent in laboratory work. Gut bacteria can make methanol. Methanol is often used in laboratory work. Commercial ice cream has propylene glycol added to it to make it soft as soon as it comes out of the freezer. Cyanide, exposure to which is typically industrial or in the course of lab work, but small amounts come from cigarette smoke and vegetables, especially those rich in glucosinolates, nitriles, and cyanogenic glycosides, as discussed in my thyroid toxins report. Anemia, which needs to be severe with a hemoglobin under 5 grams per deciliter to raise lactate to acidosis levels. Salicylates found in aspirin and in many fruits, vegetables, herbs, and spices. Metformin used to treat diabetes, and thiamine deficiency. Acid-base disorders can cause lactate to rise. An alkaline state promotes glycolysis, and since an acidic state does the opposite, acidity drives the cell to export lactate into the blood to preserve its ability to run glycolysis. So either of these deviations can raise lactate. Among the many causes of acid-base imbalances, we have vomiting causing alkalosis through the loss of stomach acid and diarrhea causing acidosis through the loss of bicarbonate. Infections, especially of the urinary tract, can raise lactate. Fermented foods and gut bacteria can be sources of lactate, although this is generally D-lactate, or at least it generally includes D-lactate. If bacteria are the source of systemic lactate, you can confirm it with a test for D-lactate because they generate a mix of L and D, and human metabolism only produces D-lactate from methylglyoxal, which I did my doctoral dissertation in, and converts it to pyruvate, but most human lactate is L-lactate. Since bacterial lactate would include both isomers and human would be overwhelmingly L, then bacterial lactate would elevate L-lactate, but you can see the footprint of that or the signature of that by looking at D-lactate. Protocols for measuring the lactate to pyruvate ratio in blood instruct the phlebotomist not to use a tourniquet for fear of the local restriction of blood flow causing a local increase in lactate production, but this is generally unnecessary under normal circumstances. Eating usually raises lactate, especially eating carbohydrate, but not that much. So it should increase it within the normal range. Some conditions in the list I just gave you are straightforward because their effects are transient. For example, lactate will be high during or soon after intense exercise, but then it'll go back to normal. This is true of shivering, seizures, states of trauma and shock, and most cases of vomiting and diarrhea. In this case, we are going to be focusing on specific abnormalities in energy metabolism that lead to chronic health problems or become unique limiters for high performance that are addressable with dietary modifications and nutritional supplements. One to narrow in on nutrition-responsive idiosyncrasies of energy metabolism. The main reasons it would be productive to seek out nutrient-responsive idiosyncrasies of energy metabolism would be the presence of a simple dietary nutrient deficiency, nutrient-responsive inborn errors of metabolism, or heterozygous carrier status for each such nutrient-responsive inborn error. Inborn errors of metabolism are rare genetic defects in metabolic pathways or structural features of cells. While each disorder is very rare on its own, collectively they number 1,451 diseases, and carrier status, heterozygosity for diseases with recessive inheritance, for one or more of them is common. Remember, I don't treat or diagnose diseases, so this is not a roadmap for diagnosing these diseases, nor is it a roadmap for treating them. And we are only concerned with learning from the treatment of these diseases to understand how we can optimize our own biochemistry for the sake of wellness. So there's basically three conditions that we want to think about. One is a nutrient deficiency or imbalance, common. One is a diagnosable inborn error of metabolism, rare, but way more common than you would think. Carrier status for an inborn error, common. Carrier status is thought to be more often than not silent symptomatically. There's various compensations. I've gone through that in great detail in my Super Unlock episode, and so I'm going to largely gloss over that here. But the gist of the issue is this, that if you're a carrier, you have 50% on average of the biochemical impairment found in the disease, and these generally lead to age-adjusted or age-delayed onsets of a shadow of the disease. As an example, heterozygous familial hypercholesterolemia does not cause a guarantee of heart attacks. If it is heterozygous, the homozygous can cause infants to have basically guaranteed heart attacks without medical treatment. The heterozygous carrier status just leads to heart disease starting to emerge at age 35 and then becoming more common with the peak increase in risk found around age 60. And then as people get old, everyone starts to be at risk of heart disease, and the gap between not having carrier status and having it begins to narrow. But the point is that between the 4th and 7th decades of life, or really between the mid-4th decade of life and the end of the 6th decade of life, the main effect is to take a shadow of the disease and cause it to emerge as a unique limiting factor in someone's longevity. There are other, there's other examples I've gone through in the Super Unlock episode and article with biotinidase, Wilson's disease, and so on. And we also want to think of the signs and symptoms of nutrient deficiencies. I'm going to gloss over this here as well because these are all outlined in great detail in my ebook, Testing Nutritional Status, the Ultimate Cheat Sheet. Now, there are also just a basic guide to when to look for inborn errors of metabolism that's included in the article. I'm going to gloss over that as well here. And so I'm going to summarize this part by just saying that what you really want to do, because the signs, symptoms, or health metrics, if you will, that will correspond to any of these things being off are so diverse, and the mixtures can produce unique health metric effects that are not necessarily textbook in the original things that make up that combination. And so I really, really think that this is something where if you are interested in optimizing your biochemistry, you simply should be measuring glucose, ketones, and lactate at home, and you should be serially testing their ratios in a lab to correlate with your home measurements. And you should then be looking at how your health metrics correspond to the home metrics and to the lab metrics. So then you can say, in me, when my feet and hands are tingling, my lactate's usually high, or when my ketones are above X despite my carbohydrate intake, this is when I'm having sleeping problems, and so on. In other words, there are so many interacting factors that the health metric effects are going to be different in each person. And so it makes sense to collect the data, look at the correlations in you, and then make an executive decision of what specifically are these going to help you optimize? Because what I'm giving you here is the general biochemical principles. I can tell you exactly the biochemistry that's being optimized, but whether that is optimizing your sleep or your resting heart rate or your cognitive performance or your mood or your neuromuscular health, etc., those are all going to be things that are unique to you. So just take the data is how I would summarize the practical import of all those possible things that I'm glossing over. Okay, here's how to view these ratios to indicate the redox status of cellular compartments. Both of these pairs act as release valves from electrons that build up in the form of NADH, which increases the ratio of NADH to NAD+. Most NADH or NAD-dependent chemical reactions will go forward or in reverse, slower or faster, depending on this ratio. Many authors write that they are used to estimate the NADH to NAD+ ratio of the cytosol and the mitochondrion, since the movement of electrons always implies a reduction, which is the addition of an electron, and an oxidation, which is a removal of an electron. They're called redox reactions, which is short for reduction-oxidation reactions. And the molecules that shift back and forth into reduced and oxidized states are called redox couples. So NADH and NAD+ constitute a redox couple, with NAD being the oxidized form and NADH being the reduced form. Many authors will refer to this as a redox status or redox state of a cellular compartment. A higher NADH to NAD+ ratio is more reduced, a lower ratio is more oxidized. In this model, the lactate to pyruvate ratio represents the redox state of the cytosol, and the beta-hydroxybutyrate to acetoacetate ratio represents the redox state of the mitochondrion. If we take these ratios to indicate the ratios of NADH to NAD+ in the cellular compartments, that's not quite correct. A lot of people say it like that, but it's better to see them as a reflection of the pressure building up within the NADH to NAD+ ratio. So this ratio, think of it like a rubber band. It's expanding with the NADH to NAD+ ratio is going higher and higher. NADH is going up, NAD+ is going down. And that's like expanding a rubber band. That's tension that's gotta be released somewhere. And so that tension gets released into the lactate to pyruvate ratio, which allows the NADH to NAD+ ratio to become more normal than it otherwise would have been. Or it gets released into the beta hydroxybutyrate to acetoacetate ratio. So these are cytosolic and mitochondrial release valves that allow the NADH to NAD+ ratio to be preserved closer to normal. But that doesn't mean that the pressure is not released in other directions. So rather, it's also the case that reactions that go forward when NAD+ is high are more likely to operate in reverse and vice versa. So you've used the release valves, but you've also released a mess because you didn't only use the release valves, right? The release valves are essentially minimizing the mess that you would make if you didn't have them. But they're not perfect, so you will make a mess in cellular metabolism by having that pressure build up too high. So when we see a high lactate to pyruvate ratio, we can infer not only that the pressure on the cytosolic NADH to NAD+ ratio has been building up, but also that most cytosolic reactions that depend on NAD+ are likely to have been slowing down or possibly even reversing. Similarly, when we see a high beta hydroxybutyrate to acetoacetate ratio, we can infer not only that the pressure on the mitochondrial ratio has been building up, but also that most mitochondrial reactions that depend on NAD+ have likely to been slowing down or even reversing. And this has two important implications. So first, when looking at biochemical panels such as amino acids and organic acids, we should be able to see reactions changing in the direction we would predict from altered pressure within the cytosolic NADH to NAD+ ratio. Second, these ratios will critically alter the rates of glycolysis and beta oxidation. If the respiratory chain is not clearing out the electrons that they're carrying, glycolysis and beta oxidation will slow. But if the cell is hungry for energy, it will need to use the two release valves to keep glycolysis and beta oxidation going. The next thing we'll discuss is that they share in common how they wind up in the urine. And this may seem out of place before discussing how they wind up in the blood, but I'm going to talk about it here because it's broadly applicable to our discussion of each individual pair that follows. In other words, when we talk about how they get in the blood, it becomes different. This is what they share in common. So this is how the kidney filters and reabsorbs these fuel molecules. To understand how lactate and pyruvate wind up in the urine, we need to consider briefly how urine forms. And if you are watching the video or you are looking at the article, you'll see a figure from the 14th edition of the Tortora and Derrickson Anatomy and Physiology textbook, which is now in its 16th edition. And it's showing you as follows. So the glomerulus is a ball-shaped capillary bed embedded in a double-walled cup of epithelial tissue known as the glomerular capsule. Blood is filtered from the capillaries into the capsule to form the glomerular filtrate, which eventually becomes the urine. We have another figure that you can find in the video or in the article that illustrates this in a little bit more detail. The membrane of the capsule has three layers. The first has large holes that allow everything through except blood cells and platelets. Thus, it allows plasma through. The second layer consists of collagen fibers and negatively charged proteoglycans that repel negatively charged plasma proteins and prevent them from going any further. The third layer consists of slits between the feet-like pedicel projections of the podocytes, which are foot cells, which are covered in a slit membrane that prevents the passage of anything larger than 0.006 to 0.007 micrometers. This filters out large proteins such as albumin, but it allows the passage of small proteins, most fuel molecules, including glucose, pyruvate, lactate, and ketones, as well as vitamins, amino acids, ammonia, urea, electrolytes, and many other small molecules. Thus, everything we're interested in, lactate, pyruvate, beta hydroxybutyrate, and acetoacetate, will move from the blood into the glomerular filtrate at this point simply because none of it is large enough to be kept in the blood on the basis of its size alone. As the glomerular filtrate passes through the renal tubule, its contents are readjusted through two processes: tubular reabsorption and tubular secretion. The first takes substances back into the blood. The second moves things out of the blood and into the filtrate. This is achieved with specific transporters of two types. Passive transporters will allow something specific to flow from an area of high concentration to an area of low concentration, and active transporters will use energy to pump things from an area of low concentration to an area of high concentration. In general, the body does not want to waste fuel molecules by losing them in the urine. However, the reabsorption of lactate, pyruvate, and ketones uses monocarboxylate transporters. These transporters do not use energy, and thus can only allow the fuels to passively flow along their concentration gradients from whichever side of the membrane they're higher on to the side on which they're lower. This means that the ability to reabsorb these molecules from the glomerular filtrate is directly dependent on their rapid transport from the glomerular capillary blood to surrounding kidney cells that will quickly burn them for energy, or in the case of lactate and pyruvate, convert them to glucose. The monocarboxylate transporters couple the transport of fuel molecules to the transport of hydrogen ions. This maintains electric neutrality in body fluids. Since hydrogen ions are responsible for acidity, these fuel molecules are less likely to be reabsorbed during acidosis, when the priority of the kidney is to allow hydrogen ions to be flushed into the urine. Additionally, all of these molecules are negatively charged and contribute to the solute load of the blood. The kidneys have to negotiate numerous priorities, including maintaining electric neutrality, maintaining acid-base balance, and regulating the solute load of the blood. Thus, generally, the kidneys will do whatever they can to avoid losing fuel in the urine. However, their ability to reabsorb fuel molecules is dependent on their ability to burn them for energy, and they may face decisions concerning acid-base balance, electric neutrality, or solute load that cause them to let a small amount of these molecules through. Nevertheless, since all four of them share the same characteristics with respect to acidity, charge, and solute load, the only thing that should impact their ratios in the urine is the state of energy metabolism within the kidney. As we will see coming soon, the kidney's state of energy metabolism differs from that of the other tissues in the body, and this can explain why urine results may differ from blood results. How lactate and pyruvate wind up in the blood. Lesson 15 of my energy metabolism class covers the reasons we convert pyruvate to lactate. It's best used in conjunction with the writings of George Brooks, who's criticized aspects of the mainstream textbook view of lactate. In brief, NAD+ takes electrons from glucose, becoming NADH. And these electrons must be removed in order to regenerate NAD+ so that glycolysis can continue to run. The electrons can be removed using the malate aspartate shuttle or by converting pyruvate to lactate. If the respiratory chain is functioning highly enough to harvest the energy in those electrons and use them to convert oxygen to water, the malate aspartate shuttle or lactate will bring the electrons from the mitochondrion for that purpose. However, if the respiratory chain is not functioning highly enough, or if the malate aspartate shuttle is not working, the electrons become stuck in the cytosol and there's nowhere for them to go except to leave the cell carried by lactate. The relative role of lactate itself versus the malate aspartate shuttle in moving the electrons into the mitochondrion is controversial, but it's clear that both paths exist and that the relative importance of each of them depends on the type of cell being studied. There are several tissues where lactate that leaves one cell is taken up to be used for energy in a nearby cell. Astrocytes feed lactate to neurons. The Sertoli cells of the testicles feed lactate to sperm. In the kidney, the medulla feeds lactate to the cortex. During exercise, fast-twitch muscle fibers feed lactate to slow-twitch muscle fibers. Gut metabolism of microbial lactate is poorly understood. Most likely, lactate-producing bacteria feed lactate to other bacteria that consume it and generate propionate, butyrate, acetoacetate, and succinate. They may also feed the lactate to intestinal cells. In the eye, the cells of the cornea and lens have low densities of mitochondria to allow a clear light path through, and they feed lactate into the anterior chamber of the eye. Whether it winds up in the blood or is oxidized within the eye is unclear. Red blood cells have no mitochondria. This makes room for hemoglobin so that they can carry oxygen. They thus rely exclusively on producing lactate to regenerate NAD+, and they have nothing that they can do with lactate or pyruvate except send them into the blood. Red blood cells act as a major buffer for excursions of blood glucose, and they take up glucose after a meal to metabolize it to pyruvate and lactate or to store it as glycogen. As a result of this, red blood cells are responsible for nearly constant drip of lactate and pyruvate into the blood and are mostly responsible for the basal levels of these substances and for their rise in response to a carbohydrate-rich meal. During sufficiently high-intensity exercise, lactate production by fast-twitch fibers exceeds the capacity of slow-twitch fibers to consume it, and this causes blood lactate to rise. The liver clears 70 to 80% of lactate from the blood, and the kidney clears most of the remainder. In both tissues, the fate of the lactate is largely to be converted to glucose, which is then returned to the blood. When this is caused by glycolytic activity in muscle, the muscle reuses the glucose to make lactate again, and this is known as the Cori cycle. The clearance of lactate by the liver and kidneys restrains the blood lactate from rising excessively and makes the rise that occurs in high-intensity exercise return to baseline very quickly. Typically at rest, blood pyruvate concentrations are 0.1 millimoles per liter, and blood lactate concentrations range between 0.5 and 1.5 millimoles per liter, yielding a normal lactate to pyruvate ratio close to 10. However, hypoxia or other disruptions to metabolism can increase lactate production by any tissue far above normal, increasing entry of lactate into the blood. If these disruptions also impact the liver and kidney, this will disrupt lactate clearance. As a consequence, blood lactate will rise above normal. How lactate and pyruvate wind up in the urine. Kidney seems to have a greater capacity than liver to oxidize lactate, and acidic conditions strongly stimulate the kidney's conversion of lactate to glucose. Thus, urine lactate may often be normal when blood lactate is elevated, and during intense exercise it raises plasma lactate to 20 millimoles per liter. Only 2% of the lactate cleared from plasma winds up in the urine. In animal experiments, artificially sustained lactic acidosis at 10 millimoles per liter causes 10 to 12% of the lactate cleared from plasma to wind up in the urine. Nevertheless, since the tubular reabsorption is dependent on the metabolic clearance of these molecules within the kidney cells to form glucose or ATP, and since the two molecules have similar impacts on other priorities of the kidney such as charge, acidity, solute load, and so on, their loss in the urine should increase if the kidney lacks sufficient energy to invest in gluconeogenesis, and their ratios should, in principle, reflect the state of the NADH to NAD+ balance within the kidney. Now that's lactate and pyruvate. This is how ketones wind up in the blood and urine. Our most detailed understanding of how different tissues handle ketogenic conditions comes from early research in obese humans subject to extended fasting. This will be the main source of what I'm about to say, along with lessons 32, 33, and 34 of my energy metabolism class. During ketogenic conditions, the liver makes ketones for the rest of the body, and during extended keto adaptation, almost entirely for the brain. The liver, therefore, bears the brunt of fatty acid oxidation for far more than its own needs for ATP and becomes buried in electrons removed from fatty acids during their partial breakdown into ketones. Thus, the mitochondrial release valve is initiated. Acetoacetate is converted to beta-hydroxybutyrate. As with the lactate release valve for glycolysis, this converts NADH to NAD+, which can then be used to keep fatty acid oxidation going. The liver releases more than 80% beta-hydroxybutyrate. Beta-hydroxybutyrate must be converted to acetoacetate using succinyl-CoA before it can be burned for energy. Succinyl-CoA must be produced from glucose or amino acids. Under conditions of carbohydrate restriction, glucose is conserved and succinyl-CoA will be largely derived from amino acids. During these conditions, the kidney is far more electron poor than the liver since it is not tasked with burning fatty acids for the rest of the body. It does, however, engage in a significant amount of gluconeogenesis from amino acids. Gluconeogenesis requires a threefold leveraged investment of ATP, operating at a major loss for the sake of the tissues that need the glucose. Because of its high rate of amino acid metabolism, the kidney has an abundance of succinyl-CoA. All of these factors position the kidney to be the prime consumers of beta-hydroxybutyrate. Studies of fasting humans suggest that the kidney consumes about 90% of the beta-hydroxybutyrate made by the liver and releases 59% of it as acetoacetate into the general circulation. This allows the kidney to earn 1.5 molecules of ATP for each molecule of beta-hydroxybutyrate that it converts to acetoacetate, which it can then invest in the expensive process of gluconeogenesis. This in turn allows other ketone-using tissues to use acetoacetate without having to worry about breaking down amino acids to get succinyl-CoA. However, during extended keto adaptation, the muscle primarily burns fatty acids for energy, saving ketones for the brain. Muscle, under these conditions, will extract acetoacetate from the circulation and release beta-hydroxybutyrate. My suspicion is that it does this because fatty acids are burned for energy in the mitochondrion, unlike glucose, which is burned for energy in the cytosol. Thus, fat-burning muscle does not have access to the lactate-based electron release valve. Moreover, since muscle is completely oxidizing the fatty acids for energy rather than making ketones, it also does not have access to the ketone-based release valve unless it extracts acetoacetate in from the blood for that purpose. Thus, while the liver initially sends ketones into the blood in a roughly 4.5:1 ratio in favor of beta-hydroxybutyrate, this proportion will go up and down as the kidney and muscle flip it back and forth to manage their own electron balance. As most ketone-using tissues preferentially take up acetoacetate, since it's easier to burn it for energy. What you see in the blood during a venous draw or a finger prick will reflect all of these processes collectively. Most likely as a result of the kidney's conversion of beta-hydroxybutyrate to acetoacetate, the urinary excretion of acetoacetate after an overnight fast is greater than that of beta-hydroxybutyrate. However, under extended keto adaptation, most tissues reduce their ketone utilization to conserve ketones for the brain. Beta-hydroxybutyrate levels in the plasma rise considerably, and a beta-hydroxybutyrate comes to strongly dominate acetoacetate in a urine ratio of about 8:1. I suspect that this reflects the fact that beta-hydroxybutyrate is now reaching the kidney at concentrations that have saturated its NAD+ reserve, and its conversion of beta-hydroxybutyrate to acetoacetate is no longer complete enough to keep the proportion of acetoacetate in urine high. Thus, ketone ratios in the urine are not expected to be the same as in the blood. However, we can still delineate normal ranges for each under particular conditions and use deviations to make inferences about the state of energy metabolism in the kidney from the urine and in the body overall from the blood. So, here are the normal ranges in blood and urine. And in this section, we're going to focus on venous blood taken from the forearm and urine, since these two samples are the ones that are practical to obtain. There's literature on samples taken from the hepatic artery, the veins of legs during exercise science studies, and cerebrospinal fluid, but we're not going to cover them because, which of us is going to go to Quest and be like, test my vein of my leg while I exercise? Normal lactate to pyruvate ratios in the blood. According to Quest, a normal blood lactate to pyruvate ratio is 10:20. According to Chapter 1, Section 4.14 and Chapter 3, Sections 1.2 and 2.2 of the Sotobre textbook, Inborn Metabolic Diseases, Diagnosis and Treatment, a normal or low lactate to pyruvate ratio is less than 12, and the ratio is regularly about 10. In a series of 11 normal children ages 1 to 7 published by Sotobre and colleagues in 1997, blood lactate to pyruvate ratios ranged from 6 to 14.5 across the day. In a series of 90 apparently normal children from Spain who were undergoing minor surgical interventions but who did not have any pathological symptoms, the ranges for the blood lactate to pyruvate ratio were 7.8 to 28 in those between the ages of 1 month and 2 years old, 8.1 to 25 in those aged 2 to 8 years old, and 7.7 to 24 in those aged 8 to 18 years old. I question whether children undergoing surgery are normal. To begin with, they could be under stress. More importantly, what's the surgery for? A biopsy of a growth, a correction of a fracture, a removal of a skin lesion? Most likely this population is biased towards children in a lower state of health than a random sample would be. Therefore, I'm inclined to give it less weight, and even though it has a larger sample size than the other studies, I want to rely more on the other studies. In a series of 72 Dutch children aged 3 to 15 hospitalized because of behavioral and somatic problems without specific neurological symptoms, the population seems similarly questionable for the generation of a normal range, but the normal ranges were more consistent with the rest of the data. The 95% normal range for the blood lactate to pyruvate ratio was 6.38 to 13.91. The very concept that children were hospitalized for behavioral problems yet had no neurological symptoms seems absurd on its face to me. Which system of the body caused the behavioral problems? Was it the urinary system? Was it the integumentary system? I dare say something may have been going on in the brain. In any case, in a series of 9 healthy men aged 19 to 33 years, the average forearm venous lactate to pyruvate ratio was 10, but the range was not described. In 12 patients with subclinical hypothyroidism, mostly women aged 24 to 63 years old, and in 10 sex and age-matched controls, the mean resting blood to lactate pyruvate ratio was approximately 9, but the range was not given. In 48 mostly Caucasian French men and women aged 30 to 60 years old, the blood lactate to pyruvate ratio ranged from 4.4 to 15.7. In a series of 76 men and 23 women in the UK, blood lactate to pyruvate ratios ranged from 5.1 to 15.4, with no difference between men and women. I did my best to sift through many hundreds of studies that come up in keyword searches and find those that give ranges for healthy subjects when blood is drawn from the forearm vein. I'm not confident it's comprehensive, but as far as I can tell, the normal range is 4 to 16 with an expected average of 10, and the Quest range is shifted too far to the high end of that. Now let's look at normal lactate to pyruvate ratios in the urine. Neither the labs nor textbooks offer ranges for urine lactate to pyruvate ratios. In my best attempt to comprehensively search the literature, I was able to find only one study that reported urinary lactate pyruvate or the lactate to pyruvate ratios in a format that would allow a description of the distribution. In 4 normal adult controls, the urinary lactate to pyruvate ratio is 5 in 2 subjects, 10 in 1, and 11 in the 4th. It's notable that these values are quite similar to those of the blood, with the average of 8 leaning a little bit more in the direction of pyruvate, as would be expected from the apparently greater NAD+ reserve of the kidney compared to the liver. In another study of 10 kidney donors between the ages of 19 and 65, the urinary excretion rates as a function of blood concentrations was reported for each individual. They tended to excrete between 0.3 and 0.8 as much lactate as pyruvate compared to what would have been predicted from their blood concentrations. It's not possible to reverse engineer the urinary concentrations from this. However, if the average plasma ratio is 10 and the average ratio of excretion rates is 0.5, then this would at least be consistent with the average ratio in urine being approximately 5. This is fairly close to my estimate of 8 from the previous study, despite both having very small sample sizes. I was unable to find any other studies with the data in the format needed for this analysis, so I would say with low confidence that a normal range for the urinary lactate to pyruvate ratio is 5 to 10. Now let's look at the normal ratios of ketones in the blood. In early experiments of fasting humans, the typical ratio of beta-hydroxybutyrate to acetoacetate starts around 2 and reaches as high as 6. However, the range of ratios were never reported. According to Chapter 1, Section 4.14 and Chapter 13, Section 1.2 of Inborn Metabolic Diseases Diagnosis and Treatment, a normal blood ratio of beta-hydroxybutyrate to acetoacetate in blood is 1 to 1.5 after an overnight fast and less than 1 after eating. However, the different authors who wrote Chapter 11 consider, in Section 1.4, the normal range of ratios to be 2.5 to 3. In a series of 11 normal children ages 1 to 7, published by Soderbergh and colleagues in 1997 mentioned above, the mean ratio after an overnight fast is reported as 1.91. Strangely, the parentheses in the table that usually include the ranges just list a single number as 2.5. All of the postprandial values are listed as less than 1. I'm guessing that this means that when the ratio was less than 1, it was usually because the beta-hydroxybutyrate was low and often below the limit of detection. So this would seem to support a normal range after an overnight fast of approximately 1 to 2.5. 2 studies from Spain examined these ratios in apparently normal children undergoing minor surgery. For the reasons I described above in the section on normal blood lactate and pyruvate ratios, I'm inclined to give the broad ranges in these 2 studies less weight. The first is the same series of 90 children that I described earlier. The complete ranges for these ratios were 0.1 to 3 in children aged 1 month to 2 years, 0.05 to 4.1 in children ages 2 to 8, and 0.05 to 2.8 in children ages 8 to 18. The second is a series of 60 children in whom 95% normal ranges for these ratios was 0.04 to 4 in children between 1 month and 7 years old, and was 0.04 to 3.5 in children between the ages of 7 and 14. In a series of 72 Dutch children aged 3 to 15 who were hospitalized because of behavioral and somatic problems without specific neurological symptoms described earlier, again, questionable for the same reasons, the bottom of the normal range, but not the top, was more consistent with the rest of the data. The 95% normal range for the blood beta-hydroxybutyrate to acetoacetate ratio was 0.85 to 8.05. The top of the range only makes sense if some of the children were in deep ketosis. In 8 men and 2 women in their mid-20s recruited for an experiment, the mean ratio was 3.2. In an earlier experiment by the same authors, possibly with some of the same subjects, in 6 men and 2 women in their mid-20s, the mean ratio was approximately 1. From a mostly Japanese language paper, the average ratio was 0.86 in 20 healthy people and was 1.31 in 29 people with hyperthyroidism. In a series of 23 men and 23 women in the UK, blood beta-hydroxybutyrate ratios range from 0.3 to 5.5 with no difference between men and women. The data for ketone ratios are much messier than the data for lactate to pyruvate ratios, and this probably reflects the fact that the ratios increase during ketosis and extended fasting, and that they reflect competing priorities of numerous different tissues that are taking up, interconverting, and releasing the ketones. Nevertheless, I'm inclined to synthesize these data as follows. The normal range for an overnight fast on a non-ketogenic diet is 0.85 to 3. On non-ketogenic diets, 0.3 to 0.8 should raise suspicions of being too low, while 3.1 to 5.5 should raise suspicions of being too high. During deep ketosis, the ratio may reach as high as 8. Regardless of diet, ratios between 0.3 or above 8 should raise strong suspicions of pointing to a metabolic abnormality. Due to the messiness of the data, I have mediocre confidence in these ranges. Now finally, let's look at the normal ketone ratios in urine. It was most difficult of all to find data on normal ranges for urinary ketone ratios. The best data comes from early, highly controlled experiments using small numbers of people. One of these showed that after an overnight fast, the ratio is between 0.65 and 2. As ketosis progressed, the beta-hydroxybutyrate would rise much faster than the acetoacetate, and the ratio reached 4. However, another early study found that the ratio rises to 8 after 38 days of fasting. In the first study, Table 6 contains 153 individual measurements taken throughout the study in 11 subjects, 7 men and 4 women. I extracted them, discarded 3 where the acetoacetate concentration was 0, and calculated the ratios of the 150 remaining measurements in a spreadsheet. In the video and in the article, you can see the distribution in a diagram. The full range is 0.1 to 11.6. The data are heavily skewed towards the lower end, meaning the mean is 1.7, the median is 1.2, and the 95% of measurements in the middle, excluding the lowest and highest 2.5%, are between 0.18 and 6.1. However, the lowest values include early ketosis, when the acetoacetate starts rising, before the beta-hydroxybutyrate takes over in deeper ketosis. Thus, I would include them in the normal range for ketosis, but not in an overnight fast. From this very limited data set, I believe the normal range for an overnight fast is 0.65 to 2, while the normal range during ketosis is 0.2 to 6, with values expected to be as high as 8 to 12 during very deep ketosis. Due to the limited data, I have low confidence in these ranges. All right, now let's talk about the stability of lactate, pyruvate, and ketones in blood and urine. And this is important because it tells us whether we can rely on the data that we get, and it tells us how to take care of the samples in the right way. So there's several stability issues that we need to be concerned with. The first is that acetoacetate and pyruvate are both keto acids, whereas lactate and beta-hydroxybutyrate are both hydroxy acids. Keto acids are more likely than hydroxy acids to spontaneously react with other substances, and they're therefore less stable. Acetoacetate also tends to slowly and spontaneously convert to acetone and carbon dioxide. The enzymes involved in their conversions, lactate dehydrogenase and beta-hydroxybutyrate dehydrogenase, both strongly favor the hydroxy acid. And so poor conversion of the samples will tend to falsely raise, thus poor preservation of the samples will tend to falsely raise both ratios. There's widespread consensus among all protocols for measuring lactate to pyruvate ratios in the blood that the concentration of these molecules must be stabilized immediately, by mixing the blood with ice-cold perchloric acid and letting it stand for 10 minutes before centrifuging. This breaks apart the cells, precipitates all of the proteins, and causes all of this material to stay at the bottom of the tube so that the liquid at the top of the tube is not serum or plasma, but rather filtrate. Lactate and pyruvate are released from blood cells, while the lactate dehydrogenase enzyme is immediately destroyed, preventing enzymatic formation of lactate during sample processing. Quest offers acetoacetate and beta-hydroxybutyrate as 2 separate tests, and neither of them call for perchloric acid precipitation. This conflicts with the protocols used by clinical labs in the investigation of inborn errors of metabolism, but it may not be a problem. The lymphocytes of rats convert acetoacetate to beta-hydroxybutyrate, and this is likely true of humans as well, but white blood cells are 1,000 times less numerous than red blood cells, and red blood cells can't make this conversion because they don't have any mitochondria. Most of the ketones in the blood should be extracellular, and separation of plasma will remove the blood cells within 10 to 15 minutes of sample collection. When unprocessed blood is kept on ice, both ketones are stable for up to 6 hours. However, if it is kept at room temperature, up to nearly half of the acetoacetate can be lost within 6 hours without any change in the beta-hydroxybutyrate. This suggests that at room temperature, blood acetoacetate tends to spontaneously degrade rather than be enzymatically converted to beta-hydroxybutyrate. In a series of 80 plasma samples, measured in duplicate with and without perchloric acid precipitation, acetoacetate concentrations were virtually identical between processing techniques. The samples were stored in a -20 freezer until analysis, and the time they were stored for was not reported. Thus, plasma ketone levels are likely to be stable as long as the blood is kept on ice before freezing and the analysis is done in a timely fashion, without any need to deproteinize the sample first. Cells are about 1 million times less numerous in urine than blood, so if the primary stability problems are enzymatic conversions in cells and spontaneous degradation of acetoacetate and pyruvate with insufficient cooling, then all of these compounds should be quite stable in urine that is properly cooled. A study using a small number of in-house samples found that both ketones are stable in urine kept in the refrigerator for at least 1 day and kept in a -20 freezer for at least 6 days. However, 24 hours of storage at room temperature made beta-hydroxybutyrate decline by 13% and made acetoacetate decline by 16 to 82%. This is consistent with the data above for blood and suggests that acetoacetate is particularly unstable at room temperature, but stable when properly chilled. In a study of 9 children with metabolic disorders, when urine was kept on dried filter paper at room temperature, it lost 20 to 25% of its pyruvate within 7 days, but the pyruvate then remained mostly stable for the next 3 weeks. At the time of finishing this analysis, I've been unable to locate any studies examining the stability of pyruvate in urine with and without perchloric acid precipitation or during storage over time at different temperatures. However, I suspect that it's stable when urine is immediately refrigerated after collection, fully frozen, and shipped with a fully frozen ice pack. The above data suggests this should also preserve the ketone bodies. Notably, when samples are processed without adequate attention to these details, this should falsely raise both ratios but should never lower them. Thus, improper sample handling could falsely mask a low ratio or give a false positive for a high ratio, but it should never be able to mask a high ratio or give a false positive for a low ratio. Now, with all the basic science and logistic concerns out of the way, here's the heart of the matter. This is how to interpret the results. If the initial observation prompting the investigation is elevated lactate, then you should go back to the list of causes of elevated lactate to consider each of those causes. And now we're going to focus specifically on nutrient-responsive impairments in energy metabolism. The first thing we're going to cover is pattern 1, which if you go back to the diagram is the red zone. Nutrient-responsive genetic causes of pattern 1. According to a 1996 paper by Sotobre and colleagues, all measurements from blood, a high lactate to pyruvate ratio, a low beta-hydroxybutyrate to acetoacetate ratio, and an abnormal rise in ketones after consuming carbohydrate are, together, strong signs of deficiencies in the enzymes pyruvate carboxylase, alpha-ketoglutarate dehydrogenase, or succinate dehydrogenase. In a small set of children with pyruvate carboxylase or alpha-ketoglutarate dehydrogenase deficiency, the blood lactate to pyruvate ratios were 26 to 37, and the blood ketone ratios were 0.27 to 0.73. They don't include values for succinate dehydrogenase deficiency. Chapter 1 of the 2022 edition of Inborn Metabolic Diseases Diagnosis and Treatment is written by Sotobre and one other author. Sotobre is the corresponding author on both this textbook chapter and the 1996 paper. In section 1.14, it says that all measurements in blood with a lactate to pyruvate ratio over 30, a beta-hydroxybutyrate to acetoacetate ratio under 1.5, and an abnormal rise of ketones in response to carbohydrate is, quote, a diagnosis of pyruvate carboxylase deficiency isolated or secondary to biotinidase deficiency or hollow carboxylase synthetase deficiency or alpha-ketoglutarate dehydrogenase deficiency is virtually certain, end quote. Thus, the claim that succinate dehydrogenase is included in this pattern seems to have been dropped, and it is not clear where it came from in the 1996 paper. Chapter 11 is written by other authors, and the section on succinate dehydrogenase deficiency does not mention these tests. The reason these enzyme deficiencies cause a high lactate to pyruvate ratio, but also the reason these enzyme deficiencies cause a high lactate to pyruvate ratio, but a low beta-hydroxybutyrate to acetoacetate ratio is because they cause a deficiency of the malate aspartate shuttle, which brings electrons from the cytosol to the mitochondrion. Because the electrons are stuck in the cytosol, the lactate levels rise. Because the mitochondrion is poor in electrons, less beta-hydroxybutyrate is produced. Malate dehydrogenase does the work of bringing electrons from the cytosol to the mitochondrion. To do this work, it requires a steady supply of oxaloacetate, glutamate, aspartate, and alpha-ketoglutarate, as well as 2 transporters to carry these substrates across the membrane, and the enzyme aspartate aminotransferase to interconvert them. Aspartate aminotransferase depends on vitamin B6. 2 other enzymes are important to keep the shuttle running. Pyruvate carboxylase supplies the oxaloacetate, and it's dependent on biotin and manganese. Alpha-ketoglutarate dehydrogenase clears away excess mitochondrial alpha-ketoglutarate, which would otherwise suppress the mitochondrial aspartate aminotransferase reaction. This enzyme is dependent on thiamine, niacin, riboflavin, and lipoic acid. Chapter 11, Section 11 of Inborn Metabolic Diseases Diagnosis and Treatment adds several more recently discovered inborn errors of metabolism that directly compromise the malate aspartate shuttle. Defects in aspartate aminotransferase, malate dehydrogenase, and the mitochondrial aspartate glutamate carrier. While it has not been demonstrated that a high lactate to pyruvate ratio and a low beta-hydroxybutyrate to acetoacetate ratio predominates in these diseases, the pathophysiology is thought to directly involve the trapping of electrons in the cytosol, and is therefore almost certain in my mind that this pattern would be found if it were looked for. Can nutrient deficiencies cause pattern 1? I believe we should be able to predict the nutritional deficiencies that would cause this pattern by juxtaposing the nutrients involved in the malate aspartate shuttle against those involved in the respiratory chain. Only the nutrients involved in the former, not those in the latter, should cause this pattern. The nutritionally relevant cofactors used in the malate aspartate shuttle are biotin, manganese, B6, the activated forms of niacin and riboflavin, and lipoic acid. The nutritionally relevant cofactors used in the respiratory chain or used for its assembly, collected from Chapter 10 of the Soderbergh textbook, are copper, sulfur, iron as heme and as iron-sulfur clusters, the activated forms of niacin, riboflavin, and pantothenic acid, S-adenosyl methionine produced during methylation, lipoic acid, and coenzyme Q10. Only biotin, B6, manganese, and thiamine are found in the first list, but not the second. As documented in the Soderbergh textbook, the biotin-responsive genetic defects in biotinidase and holocarboxylase synthetase cause this pattern. These disorders simply disrupt the supply of biotin to all biotin-dependent enzymes in a way that dietary biotin deficiency would also, so this is sufficient to make the case for biotin. However, as noted below, thiamine is also needed for pyruvate dehydrogenase, whose deficiency is responsible for the low or normal lactate to pyruvate ratio of pattern 3, which is yellow in the diagram. So, I predict that thiamine deficiency would lead to a combination of patterns 1 and 3 that I have described, that I will describe soon as the expected thiamine variant. And this is orange in the diagram. Further, manganese is also needed for the mitochondrial form of the enzyme superoxide dismutase, whose primary function is to protect the respiratory chain from the reactive oxygen species it creates during its normal function. A human defect in a transporter for manganese and zinc has been associated with defective respiratory function. Experiments in rats also suggest that pyruvate carboxylase activity is very resistant to manganese deficiency. Thus, nothing straightforward can be predicted from manganese deficiency about the balance between electron extraction and electron clearance, and nothing in descriptions of human manganese deficiency suggests useful changes in these markers. Although vitamin B6 is needed for the malate aspartate shuttle, it is also needed for glycogen metabolism and the interconversions of all the amino acids. The potential impacts from this on pyruvate, aspartate, glutamate, alpha-ketoglutarate, and malate are hard to predict. In a study comparing the concentrations of 20 different substances in the plasma of 38 patients with B6 deficiency and 41 healthy controls, lactate and pyruvate were both measured, but the only substances reported as differing were a decrease in phenylalanine and citric acid and an increase in succinic acid. Lactate and pyruvate were not reported, presumably because there was no difference. In an autistic child with combined deficiencies of thiamine and B6, thiamine brought his lactate to pyruvate ratio down from 31 to 12, yet he still had seizures that were later resolved with B6. This suggests that thiamine deficiency has massively stronger impacts on lactate than B6 deficiency, while the neurological consequences of the latter are primarily due to the inability to interconvert amino acid-based neurotransmitters such as glutamate and GABA. Thus, among straightforward nutrient deficiencies, only biotin deficiency would be expected to result in a clear picture of pattern 1. Nutritional implications for pattern 1. Pattern 1 points to several possible nutritional implications. First, biotin deficiency. See the biotin section of my Vitamins and Minerals 101 Cliff Notes for simple rules of thumb for managing biotin intake, or a much more detailed analysis in my article, High Protein, You Need More Biotin. Biotinidase or holocarboxylase synthetase deficiencies. These will look like extreme dietary biotin deficiencies and can be treated with doses of biotin at least as high as 5 to 10 milligrams per day. For a comprehensive treatment, see my article, When High Dose Biotin is Truly Needed. Pyruvate carboxylase deficiency. As described in Why High Dose Biotin is Truly Needed, pyruvate carboxylase deficiency has been for decades regarded as not biotin responsive, but this was based on trying 10 to 20 milligrams per day. The fact that an otherwise lethal mutation in holocarboxylase synthetase has been rendered relatively harmless with 1.2 grams per day of biotin makes it clear that higher doses should be tried in cases of pyruvate carboxylase deficiency. Aspartate aminotransferase deficiency. These defects have been treated with 20 milligrams per kilogram body weight of pyridoxine. I would use pyridoxal 5-phosphate or P5P instead, and 300 to 400 milligrams per kilogram body weight of serine. These were reported in children with maximum doses of 75 milligrams per day of pyridoxine. The serine is used because this deficiency indirectly compromises the NAD+ dependent production of serine. Malate dehydrogenase and mitochondrial aspartate glutamate carrier deficiencies. These are treated with ketogenic diets since these reduce glycolysis and the need for the malate aspartate shuttle. Alpha-ketoglutarate dehydrogenase deficiency is listed as having no known treatment, but it can result from reduced binding of thiamine to the enzyme. So I suspect that this may be a case where the use of high-dose thiamine has not been adequately investigated. Thus, while biotin deficiency is the only straightforward nutrient deficiency that causes pattern 1, other causes may require high-dose biotin, B6, and serine, or ketogenic diets. And my suspicion is that high-dose thiamine might be an overlooked implication. One unexplored area that I believe is likely to be relevant is heterozygous carriers for the genetic mutations described above that don't produce disease states, but they still require high doses of biotin, thiamine, or B6 carbohydrate restriction to produce optimal health. Notably, oxalate inhibits pyruvate carboxylase, and heavy metals poison lipoic acid binding sites of alpha-ketoglutarate dehydrogenase, so these can be other contributors to apparent deficiencies of that enzyme. That's pattern 1. Now let's talk about pattern 2, which is blue in the diagram. The respiratory chain of the mitochondrion harvests energy from the electrons taken from the chemical bonds in our food and uses it to make ATP. It uses the electrons to glue the 2 hydrogen atoms and oxygen together to make water. If the respiratory chain is dysfunctional, the lack of ATP will stimulate all aspects of the catabolic breakdown of food because the only way to get ATP will be to get the 2 ATP generated during glycolysis and the 1 net ATP generated by the citric acid cycle. This will lead to massive accumulation of electrons, increasing the NADH to NAD+ ratios in all the cellular components, thereby driving high ratios of lactate to pyruvate and of beta-hydroxybutyrate to acetoacetate. As of the 2022 edition of the Soderbrate textbook, there are deficits in 297 different genes that can cause respiratory chain disorders. There are hundreds of different defects in the 37 mitochondrial genes. Inheritance of mitochondrial genes is quite different than inheritance of nuclear genes. The nuclear genes have 2 copies per cell, one from the mother and one from the father. When the cell divides, each one is copied and they are divided among the 2 daughter cells evenly. Mitochondrial genes are inherited mostly from the mother, but partly from the father, sometimes, and in a way that allows hundreds of thousands of copies in each cell, not just 2. When they divide, nothing guarantees an even split. Since mitochondria divide independently, nothing ensures the proportion of alleles within a cell stays stable over time. Thus, a disorder would usually be inherited from the mother, but there could be diverse sources of normal DNA from the father, the mother's father, and so on, leading cells to have an all-mutant, all-normal, or in many cases, a mix of mutant and normal DNA. In addition, there may be many poisons of the respiratory chain. Among the many are toxic alcohols such as methanol, ethylene glycol, and diethylene glycol, the anesthetic propofol, reverse transcriptase inhibitors, cyanide, salicylates, metformin, and berberin. Ciprofloxacin appears to impair the respiratory chain by damaging mitochondrial DNA. Finally, there are many nutritionally relevant cofactors needed as components of the respiratory chain or needed for its assembly. Compiled from Chapter 10 of Soderbrate, Inborn Metabolic Diseases, Diagnosis and Treatment, these include copper, sulfur, iron as heme and iron-sulfur clusters, the activated forms of niacin, riboflavin, and pantothenic acid, S-adenosyl methionine produced during methylation and therefore dependent on the nutrients involved in methylation, lipoic acid, and coenzyme Q10. Isolated genetic deficiencies in the respiratory chain are likely to produce pattern 2. With the toxins, it's less clear because toxins can have many targets, not just the respiratory chain. For the nutritionally relevant cofactors, it is also less clear. Only CoQ10 and copper are used in the respiratory chain, but not in the catabolic breakdown of food molecules. However, copper is so integral to iron metabolism that it's likely to have secondary reverberations on the catabolic breakdown of food molecules that would be hard to predict. CoQ10 is not an essential nutrient, but it is a widely available supplement found in food and a successful treatment for some of the disorders of its synthesis. Since many of these would have unpredictable consequences on the ratios of concern, not finding pattern 2 should not be used to rule them out. If pattern 2 is found, this should point in the direction of the respiratory chain. Even if the problem's not directly caused by easy to find and remove toxins or by dietary nutrient deficiencies, the condition would likely be improved by fixing any problems on either front. As noted in the elevated lactate has many causes section, alcohol consumption can mimic pattern 2 and should be avoided for 48 hours before testing these ratios in a commercial lab. Here are the nutritional implications for pattern 2. Out of hundreds of genetic defects, 2 of them have specific successful treatments. The ACAD9 enzyme is needed to help assemble complex 1 and possibly complex 2 of the respiratory chain. And genetic defects in this enzyme are treatable with riboflavin at 20 milligrams per kilogram body weight per day, up to a max of 400 milligrams per day. The second is CoQ10, is a component of the respiratory chain that transfers electrons from complexes 1 and 2 to complex 3. Defects have been identified in 10 genes involved in its synthesis, and some but not all of them are treatable with 30 milligrams per kilogram body weight per day of CoQ10 in children and 3 grams per day in adults. A number of experimental therapies are in the pipeline. Some of these include pharmaceutical stimulators of mitochondrial biogenesis, such as benzofibrate, ovomeloxolone, REN-001, and KL1333 on the one hand, and on the other hand, rapamycin, a stimulator of mitophagy. However, some of the drugs have more generic antioxidant, NRF2, or NAD turnover targets that stimulate mitochondrial biogenesis indirectly. It also seems to me that there's widespread interest in the potential to potentiate mitochondrial biogenesis with the fasting-associated PGC1 alpha pathway without sufficient cognizance of the need to cyclically step on the fed state-associated mTOR pathway to fuel fresh mitochondrial growth. My fasting feeding reset protocol is based on cyclical promotion of mitophagy in the fasting state and mitochondrial biogenesis in the fed state. Mitochondrial biogenesis is potentiated by the fasting state signaling and fueled by the fed state signaling. I suspect this would be most helpful for situations where the mitochondrial DNA have been damaged, such as with ciprofloxacin. Conceivably, it might have benefit in people who have very heterogeneous mitochondrial DNA, where cells may choose to selectively eliminate the mitochondria with more defective DNA and regenerate fresh mitochondria selectively from the more healthy ones. In Chapter 8 of his book, Eat for Energy, Ari Whitten, with Alex Leaf, collected numerous studies together of supplements that have been shown to improve mitochondrial health generally, without respect to whether the specific causes of poor mitochondrial function relate to oxidation of food molecules or the respiratory chain. One of them is specifically worth noting here. PQQ stimulates mitochondrial biogenesis through means that would place it in the fasting portion of my fasting feeding reset due to its opposition to mTOR and promotion of PGC1 alpha. To the extent it may prove useful, the dose that has been studied in humans is 20 milligrams per day for 8 weeks. Most of the others, essential nutrients, phospholipids, amino acids, astaxanthin, curcumin, D-ribose, forskolin, gynostemma, and ginseng, have diverse effects across the catabolic breakdown of food molecules and the respiratory chain, or on fasting and feeding state signaling, and so therefore may be of value to many people, but do not specifically fit into this model of how to interpret changes in the ratios under discussion. Creatine is notable for its specific role in improving the efficiency of cytosolic ATP, and so it may take stress out of the lactate release valve. Acetyl L-carnitine is notable for its specific promotion of fatty acid oxidation, which could put stress on the riboflavin requirement when riboflavin is inadequate, or with adequate riboflavin could put stress on the respiratory chain when the respiratory chain is inadequate. We can brainstorm further rational strategies for respiratory chain support by thinking about the respiratory chain in more detail. The respiratory chain has 4 complexes through which electrons flow, and a 5th that synthesizes ATP. CoQ10 carries electrons between the first 2 complexes and the 3rd. Cytochrome C carries them between the 3rd and the 4th. The mitoswab test can distinguish between defects in different complexes of the respiratory chain. However, it cannot determine whether the cause of decreased activity is nutritional, toxic, or genetic. Therefore, I would run nutritional testing to fix any deficiencies of nutrients involved in the respiratory chain before I would run this test. Carbohydrates disproportionately feed into the first complex, while fats disproportionately feed into the second. These are differences of proportion, and it is not possible to avoid using either complex. However, it suggests that ketogenic diets would be more suitable for complex 1 disorders, and that low-fat diets would be more suitable for complex 2 disorders. There are some cases documented of improvement on ketogenic diets. Infrared light in the 700 to 1000 nanometer range can penetrate tissues and directly provide energy to complex 4. Use of a sauna with infrared in this wavelength may help with disorders of complexes 1, 2, or 3. Methylene blue may help for complex 1, 2, or 3 disorders, or 4, but methylene blue is a highly nonspecific and just generally rewires the respiratory chain along the path of least resistance. And so its main benefit is that when there is an impairment, it can create a new path of least resistance that is better than the impaired alternative. I have a much more detailed article, video, PDF, and podcast on methylene blue, so check that out for more details on this. But generally speaking, methylene blue should be in the list of things to try, but should come after things that are more natural and have more specific effects, such as the ones that I just discussed. Several other strategies may be helpful in complex 3 disorders. One case report treated someone with a complex 3 disorder with 10 milligrams metadione and 1 gram of vitamin C every 6 hours on the hypothesis that this would bypass complex 3 and provide electrons from vitamin C to metadione to complex 4. I would prefer to use MK4 rather than metadione, since MK4 is naturally present in mitochondria and has similar redox properties as metadione. Both of these are forms of vitamin K. A second case report used this treatment and found that it did not normalize metabolism in muscle, but it did normalize metabolism in the brain. Copper is distinguished among the nutritional cofactors for only being involved in complex 4. This makes copper the only essential nutrient within the context of fuel metabolism, is highly specialized both to the respiratory chain and to a specific complex within the respiratory chain. There's no evidence that supraphysiological doses of copper would help complex 4 disorders, and they may in fact hurt by causing oxidative stress. However, an apparently isolated defect in complex 4 could conceivably result from a dietary copper deficiency or defect in copper transport. So that's pattern 2. Now let's consider the last pattern, pattern 3, which is yellow in the diagram. The third pattern is high pyruvate with a normal or low lactate to pyruvate ratio. Lactate is usually high, but doesn't have to be. This suggests pyruvate dehydrogenase deficiency. The 1996 Soderberg paper shows pyruvate dehydrogenase deficiency elevating lactate to the 2.4 to 4.6 millimoles per liter range, and pyruvate mildly elevated to 0.21 to 0.45 instead of 0.08 to 0.21, but with a normal lactate to pyruvate ratio of 9.1 to 11.5. In a case report of a 7-year-old boy, his resting lactate was elevated at 3.7 millimoles per liter, and his pyruvate was elevated at 0.4 millimoles per liter, but his lactate to pyruvate ratio was normal at 9.2. However, an exercise test drove his lactate to pyruvate ratio only up to 24.2, whereas controls doing the same exercise had their ratio shoot to 35. One major difference was controls had their pyruvate shoot up but stay within the normal resting range at 0.18, whereas his started high and rose even further to 0.54. The exercise test seems deeply impractical, as it would be hard to generalize from the procedures used for male children to any other demographic, and hard to carry out with any reliability. However, high pyruvate along with high lactate is the distinguishing feature of the pattern. Normal fasting pyruvate is 0.17 millimoles or less per liter. Even after eating or exercising, it doesn't raise higher than 0.21 millimoles per liter. In pyruvate dehydrogenase deficiency, it starts high and goes even higher with exercise. Pyruvate dehydrogenase is dependent on thiamine, lipoic acid, and the activated forms of riboflavin and niacin. It converts pyruvate to acetyl-CoA and is every bit as essential as glycolysis to be able to derive energy from carbohydrate. It is also every bit as irrelevant as glycolysis for the ability to derive energy from fat. 500 to 2,000 milligrams per day of thiamine and ketogenic diets are used to treat genetic disorders in pyruvate dehydrogenase. The ketogenic diet minimizes the need for the enzyme, and the thiamine helps correct deficiencies in thiamine binding to help the enzyme function correctly. Notably, heavy metals poison the lipoic acid binding site of alpha-ketoglutarate dehydrogenase. A thiamine deficiency is not likely to recreate pattern 3 per se, but I would expect it to create a mix of patterns 1 and 3, which I described below. The expected thiamine variant, orange. While thiamine is necessary for alpha-ketoglutarate dehydrogenase, and thus necessary for the malate aspartate shuttle, It is also needed for pyruvate dehydrogenase, the enzyme that converts pyruvate to acetyl-CoA. As I already described, deficient pyruvate dehydrogenase can block pyruvate metabolism and raise pyruvate levels. As noted in Testing Nutritional Status, the Ultimate Cheat Sheet, the most telltale pattern for dietary thiamine deficiency is a combined elevation of pyruvate, lactate, alanine, and glutamate. A low lactate to pyruvate ratio has been found in the cerebrospinal fluid of thiamine biotin responsive basal ganglia disease, which appears to be driven by impaired thiamine transport into cells. On the other hand, an autistic child with combined deficiencies of thiamine and B6, thiamine alone brought his lactate to pyruvate ratio down from 31 to 12. I believe a simple dietary thiamine deficiency or any defect in thiamine handling that broadly impacts all thiamine-dependent enzymes is likely to cause a combination of patterns 1 and 3. Thus, lactate and pyruvate will both be elevated with no clear prediction of their ratio. It could be high, low, or normal. On the other hand, the pattern 1 red characteristic for a low ketone ratio should predominate because thiamine is needed for the malate aspartate shuttle due to its role in alpha-ketoglutarate dehydrogenase. This implies that, like biotin, a lack of thiamine could mask a deficiency in the respiratory chain. And in that context, an excess of thiamine might create an excessive burden on the respiratory chain that might be resolved with respiratory chain nutrients, especially riboflavin or CoQ10. 4 ways the patterns can combine. Consider now that we have discussed 3 primary defects, just as there are 3 primary colors. We have defects in the malate aspartate shuttle, red, defects in the respiratory chain, blue, and defects in pyruvate dehydrogenase, yellow. As a thought experiment, what would our 3 secondary colors be? What happens when we mix these defects together? The combination of patterns 1 and 2 makes purple. This should lead to a masking of pattern 2. It manifests as pattern 1 at first, but if you correct the malate aspartate shuttle, suddenly pattern 2 manifests. Example, me. I had pattern 1 until I corrected it by supplementing biotin, then pattern 2 emerged, which needed to be fixed with riboflavin. The combination of patterns 1 and 3, orange, is thiamine deficiency. It's what I described above in the expected thiamine variant. The high lactate to pyruvate ratio and low ketone ratio of pattern 1 has high pyruvate added to it. That neutralizes the lactate to pyruvate ratio but keeps high levels of both. Thus, high lactate, high pyruvate, normal or unpredicted ratio, and low ketone ratio are what you find in the orange pattern of thiamine deficiency. And then the combination of patterns 2 and 3 makes green. This should take the high lactate to pyruvate ratio and high ketone ratio of pattern 2 and add high pyruvate to it, neutralizing the ratio. Thus, it looks like the previous combination, high lactate, high pyruvate, normal or unpredictable ratio, but the ketone ratio is high rather than low. This is what you might get if you're heterozygous for pyruvate dehydrogenase deficiency and for a respiratory chain disorder. Now suppose we combine all 3 to make the brown variant. For example, take someone with a moderate respiratory chain disorder, pattern 2 or blue, who's otherwise asymptomatic and now introduce a thiamine deficiency. The thiamine deficiency adds patterns 1 and 3, red and yellow. This should lead to high lactate, high pyruvate, a normal or unpredictable ratio, and it should mask the respiratory chain disorder until the malate aspartate shuttle is fixed. So just as I unmasked my respiratory chain disorder with biotin, this hypothetical person would unmask their respiratory chain disorder with thiamine. At first, the problem is brown. By removing red and yellow from the mix, one can finally see the blue, and now one can understand that blue, respiratory chain, nutrients may be needed in the balance. Imbalances between nutrients. If we apply the nutrients to the 4 pattern combinations discussed in the last section, we arrive at the diagram that's available in the video and in the article. You can use this going forward to plan a nutritional intervention or going backward to help explain imbalances that you or someone you know experienced without being able to explain them. Having lactate to pyruvate and ketone ratios would greatly help to substantiate the appearance of an imbalance with an objective measure. It also helps calibrate the use of home measurements of glucose, beta-hydroxybutyrate, and lactate for real-time monitoring of an intervention. The principle of the diagram is this. If you move from the brown center to any of the outer areas rather than into the normal ranges, or if you move from the purple, orange, or green zones into the red, yellow, or blue zones rather than into the normal ranges, you have likely created or unmasked a need other than the one being addressed. For example, I thought I was in the red zone, used a red nutrient, biotin, to take me to normal, but it instead took me to blue, showing me that I had initially been in the purple zone, not the red. In my case, I needed to balance biotin and riboflavin because of the specific genetic mutations I identified with whole genome sequencing. Someone else, and as I told you at the beginning, I have updated this now to realize that there are better strategies than fixing the biotin problem for me, but we'll save the details for another time. Someone else could have experienced the exact same thing from supplementing thiamine or B6 without addressing their unknown needs for CoQ10 or copper. Or, unfortunately, they may have unmasked one of the many respiratory chain disorders that don't have simple curative treatments. Alternatively, another person might think that they're in the red zone and need biotin, but this moves them to the yellow zone because they also needed thiamine, showing all along that they had actually been in the orange zone. There are many different permutations, so I'll leave it to the figure to act as a cheat code that can be used on an as-needed basis to help you think about your own case or that of someone you're trying to help. Best way to have access to that figure permanently so you can use it as a cheat code is to sign up for the MasterPass so you have enduring access to the article. Home measurements. When specialists in inborn errors of metabolism are trying to narrow in on these and several related diseases in energy metabolism, one of the tests they'll do is they keep someone for an entire day with an indwelling venous catheter flushed in saline, and they measure their glucose, fatty acids, lactate, pyruvate, ketones, and free. When specialists in inborn errors of metabolism are trying to narrow in on these and several related diseases of energy metabolism, one of the tests they'll do is they keep someone for an entire day with an indwelling venous catheter flushed in saline and measure their glucose, fatty acids, lactate, pyruvate, and ketones at waking and before and after each meal, as well as before and during a glucose tolerance test. I did something approximating this in my self-experiments in the biochemically unoptimized state, but without being able to test the ratios. There's no home measurement for blood acetoacetate or blood or urine pyruvate, and the lack of precision in look at the color urinary acetoacetate sticks makes them severely limited in their utility. Now that I've also discovered that if I'm taking high-dose riboflavin, if it makes the urine neon, it really makes them totally useless. Home measurements can reveal particular abnormalities without looking at the ratios. For example, ketones should mildly elevate after an overnight fast to 0.1 or 0.2 millimoles per liter and be suppressed by meals. Unless you're on a keto diet, they can go up to 1 to 5 millimoles per liter. Glucose should be 80s or 90s, maybe high 70s or mid 70s after an overnight fast and conform to a typical glucose tolerance curve upon eating. Lactate should be on the lower end of the 0.5 to 1.5 millimoles per liter range when fasting and rise toward the higher end after eating, especially after eating carbohydrate. Anything operating backwards, such as lactate increasing in the fasting state and being decreased by meals, a short 5 to 15 minute rise in ketones after eating carbohydrate, abnormally high ketones on a non-ketogenic diet, glucose markedly out of the range, or lactate being increased more by fat instead of carbohydrate, could point in the direction of some of the abnormalities discussed herein. For example, lactate being high in the fasting state and suppressed by carbohydrate can happen in disorders of gluconeogenesis when glucose should be low in the fasting state, or in disorders of the red nutrients discussed in this episode. Ketones rising in response to carbohydrate for 5 to 15 minutes can point toward disorders of the red nutrients, but can also occur in the blue nutrients. Interpreting these home measurements without the ratios is difficult and involves educated guesses. For this reason, I strongly advocate collecting baseline calibration of the home measurements with Quest and Great Plains blood ratios and ketone ratios, and repeating this at major milestones on new supplemental regimens. Putting it all together, interest in pursuing the strategies discussed in this post should begin with one of 4 starting points. Lactate's higher than normal, consideration has been given to everything, and elevated lactate has many causes, and an abnormality of energy metabolism is still suspected. Symptom clusters found anywhere in the range of inborn errors of metabolism to heterozygous cases to nutrient deficiencies lead you to suspect one of those things. Previous organic acid analyses upon reinspection show alterations in these ratios that seem worth investigating. Or you're just trying to optimize for performance and longevity, and you've picked all the low-hanging fruit and then want to consider it worth your energy and time to do more testing to do these things. And in fact, now in the BioOpt Health program, I've taken the approach of comprehensively testing these things right off the bat alongside biochemical data and genomic data, because I've realized that if you wait for a reason to run these tests, you wind up dragging out the solutions to issues or the keys to optimizing your biochemistry for way too long, and we really shouldn't be waiting that long. So I actually think it's better to just run these things up front. When you want to investigate specifically these ratios, you want the Great Plains Organic Acid Test, the Quest acetoacetate and beta-hydroxybutyrate, and the Quest lactate filtrate. And you can measure D-lactate simultaneously if you're concerned about bacterial sources, but I don't think it's necessary. Home tests are Nova Biomedical Lactate Plus and Keto Mojo for finger prick, glucose, ketones, and lactate. And the Testing Nutritional Status, the ultimate cheat sheet, can be used for comprehensively assessing nutritional status. If you want the full suite of genomic and biochemical data with my analysis, you can sign up for the BioOpt Health program at bioopthealth.com. Please remember that I'm not a medical doctor. This is not medical advice. I do not diagnose or treat patients. If you're in an emergency, seek medical treatment, and before you implement anything in this article, please discuss it with your physician. Under all circumstances, the proof is in the pudding. Does this model lead you to better health or better performance? Does it at least optimize your markers and not leave you with any unsolved problems? That it works will be the confirmation that the model was what you needed. And if this model helps you realize anything useful about your current or past experience right off the bat, head over to the Substack article where the link is in the description and let me know in the comments.